Hi everyone. My name is Megan and I am an instructor at the DNA Learning Center. Welcome to DNALC Live. If you're joining us again, welcome back. Today what we're going to be doing is we are going to be creating a cell model today. We call this Baggie cell. Specifically, we're going to be making a model of an animal cell. So before we can go ahead and get started with that, which by the way is something that you can actually join us in doing, we're going to be using all different types of products that you can find either in your kitchen or just around the house. So if you'd like to rewatch this video after you find some items and join me in making a model, that would be great. So before we start creating a model of a cell, let's make sure that we understand what cells are and what their function is. So all living things are made up of cells. They're basically the building blocks of life. I'm made up of all cells. You're made up of cells. The trees outside are made up of cells. My dog is made up of cells. And even my breakfast was made of cells. So there's all different types of cells. Two that we tend to focus on are plant and animal cells. But today we are going to be focusing on animal cells. Now, you and I, are we considered animals? Are we made up of animal cells? We are. So this is going to be a general version of what you would see as our cells would look, okay? So we are going to discuss some of the different types of cells that we have, and we're gonna discuss a concept called forms, fits, function. All right, so I'm gonna share my screen with you, and give me one moment. Okay, so what we're going to take a look at here is some of the different cells that you would find in us. Okay, we have things like nerve cells, we have muscle cells, we have bone cells, blood cells, all different things. Now, if you're taking a look, you're probably noticing that all of these different cells have different shapes to them. We call that its form. And what we usually say when we say form fits function, we're talking about its shape matches the job that that specific cell has. Because if you think about it, do you think that my brain cells look like my skin cells and the skin cells look like my muscle cells? No, they're probably not going to look the same because they all have different jobs. So one that I like to look at right off the bat is the muscle cells. Now we have three different types of muscle cells, but all of them have this common feature where they're kind of stretched out pretty long. That's their shape, that's their form. Now, why do you think a muscle would need to have that shape to it? What's the purpose of our muscles? What do we need to do with our muscles? We need to be able to flex them, and we need to be able to stretch them. So therefore, our cells need to then be able to do the same thing. They need to be able to stretch out as well. Now let's compare that to what we see on our next slide. These are bone cells. Now they look kind of different than the muscle cells we were just taking a look at. Well, that's because they have a different job. The bone cells tend to be much more rigid. All right. Now, why would our bone cells need to be rigid? Well, because the purpose of the bones in the body is to not only provide support, but also to protect the organs within inside the body as well. So do you think it would be good if our bones were made up of the same type of cells as muscle cells? No, that wouldn't be good. So our bone cells have a different form to them as well. Let's take a look at another one. So these are neurons, nerve cells. Now, as you can see here, they kind of stretch out really far. Well, think about the function of a nerve cell. The nerve cells need to be able to transmit messages from one part of the body up to the brain and then possibly back to another part of the body. Think about if you were to put your hand on a hot stove. That message is going to have to be transmitted through all of these different nerve cells all the way up to your brain. And then a message is going to have to be sent back down to the muscle so that you jerk your hand away. These messages need to be sent very quickly, so that makes sense why we're seeing that its form, its shape, is reaching out really far. They want to be able to reach out to that next nerve cell so that they can send the message. All right, and then my personal favorite, the red blood cells. Now these have this round shape to them, kind of dips down in the middle, almost looks like a donut. But does anybody know the function of a red blood cell? So the function of a red blood cell is to carry oxygen throughout the body. I almost think of the red blood cells as if you've ever been to a water park and you've been on the lazy river, you sit on a tube that kind of looks like that. And as you're cruising down the lazy river, you're going to bump into the sides, but you bounce right off. Or you bump into someone else and you bounce right off of them. Do you think that that would be possible both in that scenario at the water park or in the bloodstream? Do you think that would be possible if these were shaped as squares? Probably not. Or in the case of sickle cell anemia, if these were shaped kind of like crescent moons? No, they wouldn't be able to flow through the blood as well, making it harder for oxygen to get throughout the body. And also they could get caught up on one another, which could cause a clot. All right. So now that we've taken a look at some of the different types of cells and why they have their form, let's take a look at just a generic cell and talk about some of the parts within that cell. So if we think of our body as a whole, we have organs inside of our body. And each one of those organs has a different job in helping keep us alive. Well, the cell has all these different parts inside of it called organelles. And each organelle has a different job in helping keep the cell alive. So it's the same idea, just on a microscopic level. So let's talk about some of those different organelles that make up the cell. So the first one we want to talk about is right down here at the bottom, the cell membrane. So the cell membrane is the very outermost layer of the animal cell, and it provides it with protection, but also it's semi-permeable, which means that things can leave the cell or certain things can come into the cell. I almost think of the cell membrane as like my house. I'm protected inside my house, but I can leave through the door if I need to or come back inside. All right, now let's take a look inside of the cell. So all of these different organelles that we're seeing here are floating around in something called cytoplasm. Cytoplasm is a jelly-like substance, and it makes movement possible throughout the cell. As we're going to talk about it in a bit, things are going to need to be able to move around, whether it's nutrients, proteins, they need to move from one organelle to the other. So that jelly-like substance called the cytoplasm is going to help make that possible. Now, a very well-known organelle within the cell is called the nucleus. Some people like to call the nucleus the brain of the cell. Well, there's a reason for that. The reason the nucleus is called the brain of the cell is because inside of the nucleus, there's something called DNA. And if you've been joining us over the past few weeks, you have heard us talk about DNA a bunch. So DNA stands for deoxyribonucleic acid. And basically it is the instructions on how to make that specific living thing. So that's why we consider it the brain of the cell. It has all the instructions on how to make that living thing. Now, if we were to go inside of one of our cells and take out the DNA from inside the nucleus and hold it up, does anybody know how long it would be? It would be 6 feet long. I'm not 6 feet tall, but the instructions on how to make me are 6 feet long. But we'll talk a little bit, we'll talk in a little bit why it's able to not only fit inside the cell, but specifically fit inside of the nucleus. All right, so we have some other parts here that we're seeing. So I like to focus on these little dots that you're seeing around, these little red dots all over the cell and also on this part here and here. Those are called ribosomes. Ribosomes have a very important job of reading RNA and creating proteins that are necessary for survival of the cell and just the organism in general. Now, we're seeing a lot of ribosomes on one of these organelles in here, right here and here, and that is called the rough endoplasmic reticulum. I always think that's a fun word to say, the endoplasmic reticulum. So we have the rough endoplasmic reticulum and then the smooth. All right, these are basically like passageways, but we consider this one rough because it's covered in those ribosomes. Okay, another one that's quite commonly known is these that you're seeing here. They're kind of shaped like kidney beans, and those are called the mitochondria. Some people like to call them the powerhouse of the cell. The reason why they're called the powerhouse of the cell is because they create energy for the cell. All right, we have another one right here. This one is called the Golgi body. Okay, some people call it the Golgi apparatus. And I like to think of this one as almost UPS or FedEx because it helps package up different proteins and get them ready to be sent outside of the cell or to other parts within the cell. We have two others that I'd like to discuss. So we have these strands, which we call microtubules, which help support the cell and give it its shape. And then we have these purple spots that we're seeing here, which are called lysosomes. They're basically the garbage disposal of the cell. Just like how in our body, we don't want waste to build up. It's the same thing with the cell. We don't want waste to build up within the cell. So the lysosomes are going to help take care of any waste that builds up. Now, if you've joined us through our website, there's a resources tab where you can actually download this if you'd like to have it. And it also includes some of the explanations of what some of those organelles are. Now, when talking about the cell, we're going to compare it to a factory because the cell operates in a way that's very similar to the way a factory needs to operate. Now, I'm sure you've seen factories on TV or movies. Maybe you have actually visited a factory. I've heard of people going to the Crayola factory. Some people I know have gone to the Ben and Jerry's factory. And then my personal favorite was the Hershey's factory. So I like to do a comparison of the cell to a chocolate factory. And if you've ever seen Charlie and the Chocolate Factory, you've got a pretty good idea as to what a chocolate factory or what goes on within a chocolate factory. Maybe not everything though. All right, so let's think for a moment, what are some of the parts of a factory? What is going to be necessary within the factory in order for us to get that end product, the chocolate? So let's think about that for just a moment. What do we think we need? Now you can enter that into the chat box. There's lots of different things that go into a factory. So if yours doesn't make the list, that's totally okay. We can only fit so many. So the first one I'm going to put here is ingredients. In order for us to make chocolate, we need the ingredients to make chocolate, things like cocoa beans and milk. All right, so ingredients is one thing that's necessary. What would be another thing we need in order to run a chocolate factory? We're going to need things like machines. And within that machine category, I also consider the employees because somebody needs to be able to work those machines. All right, so the machines are going to do different jobs within the factory in order to either create the chocolate or package it up and get it ready to be sent out. What else? Those employees, do they just come in every day and know exactly what to do? Sometimes, but you know what? There's usually a boss. There's going to be a boss who's going to give instructions so that everybody within the factory knows what they're doing. Now, while we're on the topic of the boss, does the boss stay down where the machines are? No, the boss is going to have an office. That's another part of our factory. All right, what else do we need? So are all these machines going to be outside? What do you think? Give me a moment. No, they're not going to be outside. We need a building. The actual factory building, okay? It's gonna provide protection. We don't want it outside, out in the weather. So we are going to include a building. All right. Now, in order for all those machines to function and work, there's something else that needs to be running throughout the factory. It's going to be electricity. We need a power supply. It's going to run over into our next box, but that's okay. Power supply. And the last one is a little tricky, but I think it's so important that there's some sort of ventilation system within the factory. It's going to get really hot in there with all those people and all those machines running. So it's important to have some sort of work environment that allows the air to move around, allows the people to move around. So I usually just put it as like an air conditioning system. All right, so now what we're going to do is we're going to take those parts of the factory and see how they can relate to the parts of the cell. So just think about it for a moment. Take a look at these different parts and let me know if there's one part that you think you know is going to be as the part of the cell. All right, so I usually start off with, let's take a look. The boss. The boss is pretty easy to start off with because we know that there's a part of the cell that's kind of in charge. Now, a lot of people jump to say that this is the nucleus, but think about it. The nucleus is where what is held? The DNA. So the boss is actually considered the DNA, and we know that the DNA is stored inside of the nucleus. So that's going to be the boss's office. All right, good. Now let's jump down to power. Now there was one organelle within the cell that I said was called the powerhouse. Which one was that? That is going to be the mitochondria. Mitochondria. All right. And then the building, that one's a little easier as well. The building, what is going to be the very outside part of the cell? Because the building is the very outside part of the factory. It's going to be the cell membrane. Okay. And then we have 3 more. So the machines one, this one's a little tricky, but remember, there's all different machines. They all have a different job. And I just consider this all the other organelles. We've named some of the organelles, like the nucleus, the mitochondria, but all of those different machines, all of those different organelles. Okay. Our last one down here, so the environment that everything is in. What was that substance that all of the organelles kind of float around within? What was that called again? The cytoplasm. All right, and then this last one up here, the nutrients, the ingredients, I kind of gave it away, but I consider that the nutrients within the cell. All right, it's looking good. So now we want to compare that to the model. We are going to be creating that model now. So I'm going to come back to this in a moment, towards the end, and we're going to go over this and kind of fill this in for you. All right, so I'm just going to just give me a moment to get all settled so that you guys can see what we are going to be working with. Okay. All right, so actually, you know what? Let me unshare my screen. That would be good. Okay, all right. There we go. Okay, so we have a whole bunch of different items that we're going to be working with today. Hopefully you'll be able to find some of these things either in your kitchen or just around your house, and hopefully you'll be able to join in making a model of a cell. So some of those items that you're going to need. So I have an array of beans here. I actually call it the bean buffet. So we have kidney beans, these green beans, navy beans, black beans, pinto beans, and then at the end, I have little pieces of spaghetti. All right. Now, if you don't have this wide variety of beans in your house, that's totally fine. You can use things like jelly beans, M&Ms, anything that you can find around the house that just has a variety of either colors or shapes. All right, another thing you're going to need is gelatin. Now, if you don't have just plain gelatin lying around, I haven't tried it yet, but I wonder if you'd be able to use a packet of Jell-O. That might be a good science experiment to try out to see if this would work with just a packet of typical Jell-O. But this is gelatin right here. Another thing you're going to need is a cup. I'm going to be using a science beaker today just so you can see because it's clear, but any kind of cup will work for this. You're going to need a plastic bag. So a Ziploc is fine, or a sandwich bag, anything like that. We're going to need string. And then you'll also need some kind of container. So I use this little plastic ball that opens up. Maybe you have one from like a gumball machine. Maybe you have one of those plastic eggs lying around. That works as well. Anything where you can kind of just close it up and close something inside. All right, so let's get started. So what we're first going to do is we are going to be using this plastic bag, like one of the parts of the cell. And this is going to be the very outside of the cell, which means it is going to be the cell membrane. All right, so the way I get this ready to go is I take my plastic bag, put my hand inside of it, and I actually just push it down inside of my cup. I take the extra and I kind of just fold it over. It's almost like you're putting a garbage bag in the trash bin, okay? Just folding that over. All right, excellent. So we have our cell membrane ready to go. All right. So now let's talk about the string. What part of the cell do you think the string represents? Now I have a lot of string here. It's a little bit tangled, but it may be 6 feet of string. Okay, what do you think is this representing? If you said DNA, you're correct. All right, so the DNA is the string. And where does the DNA go within the cell? It's going to go in the nucleus. And our little plastic ball is going to represent the nucleus. So what we're going to do is take that string and we're going to kind of coil it up, kind of roll it up so that it's going to be able to fit inside of the nucleus. All right, so it's all coiled up, kind of just like our DNA does. I place it inside of the plastic ball, snap it closed, and then the nucleus can go right inside of your cell membrane. All right, good. Now, what we're going to do is we're going to use all these different beans as the different organelles within the cell. Remember, they all have different shapes, they all do different jobs, but we're going to point out a few of them to be specific ones. Now, remember, I said there was a specific one that looks like a kidney bean, and that's going to be our mitochondria. So we're going to say the kidney beans are the mitochondria. These little tiny green peas, we're going to say those are the ribosomes, and then all the other ones can kind of represent the different parts as well. So what I do is I take my plate and I just take a pinch of each one of the beans. Okay, I like to add extra of the ribosomes, navy beans, black beans, and then I even take a pinch of the spaghetti. Now, we don't want these to kind of puncture the cell membrane, so I always take them and I break them up even smaller. So just kind of crack them into smaller pieces. All right, excellent. So now we have all of the organelles that can go inside of our cell. So I just take that and fold it like a taco and pour them in. All right, excellent. Good, so we've got our DNA inside of the nucleus. We have all the organelles, including ribosomes and mitochondria. We have the cell membrane, and now we want to add something that's going to give our cell the feeling of cytoplasm, so that jelly-like substance, and that's where the gelatin is going to come into handy. So all I do is I just open this up, and I'm going to pour some inside. All right, we'll do a little extra for fun. Okay, all right. So that's going to help us get that jelly-like feel that the cytoplasm has. Now, what do I need to add to this in order to have it have that feel? We need to add water. So I'm just going to step over to the sink, and what I'm going to do is I'm going to fill this all the way to the top, not overflow it, but really fill it up, okay? So. Okay, so now it is filled up pretty close to the top. So what we're going to do at this point is I'm going to bring the bag up carefully. Be gentle at this point. We don't want to pop the cell. I'm going to carefully lift it out of the cup. All right, excellent. Make sure we have no leaking. Looks good thus far. And what we can see here is we see that the nucleus is floating on top. That's okay, that's because there's air inside of there right now. But we see our different organelles down at the bottom, and it kind of has this cloudy look to it because of the gelatin. But if you look carefully, there's like a big clump of it. We don't want that. We want to break up the gelatin so that it's evenly dispersed throughout the water. So what you'll do at that point is you're just going to take and kind of pinch the bag very gently. Remember, there's spaghetti in there, so we don't want to go crazy. But you want to mix up that gelatin. Once you see that all of the chunks are gone, then you're done with that part. Now we want to tie this so that it doesn't spill everywhere. Do you think I should tie it up at the top? Like this? No, that's not going to be good because once I put my cell down, it's going to look like a pizza. And if I pull it back here, it's just going to flatten out like a pizza. We don't want that. So what we're going to do is we're going to try our very best to tie it all the way down at the bottom. Now this is a little tricky. You may need to ask an adult to help you, but what I do is I just twist this part. I go around and right through that loop. And again, the key is to try and make that knot as close to the bottom as possible. All right, so now we can see that the cell has a better shape to it. We can see all those different organelles in there. We can shake it up a little bit so they're not all at the bottom. All right, excellent. Now, if you have a bag like mine, sometimes I like to take mine and kind of double bag it just to make sure there's no leaks. If you do have a leak, you can just take it, put it in another bag. That's it, it's no big deal. Now, if you've ever made Jell-O before, you know that in order for it to take on that form, that jelly form, you need to take it and place it in the refrigerator. So I've done that with one of mine. So we'll swap these. I'll take this one. I'm going to put it in the refrigerator. And since I wanted to speed up the process, I put one of mine in the freezer. Okay, so now this is the cell that I made before. So we can see that it's keeping its shape much better, but it still has that jelly-like substance, like it still moves. It's not completely solid. All right, excellent. I can see all of those different organelles in there. I know that it has this jelly-like feel to it because that's representing the cytoplasm. Luckily, my cell membrane stayed intact, so I don't have any leaks right now. And what can happen is once you've done this, and if you have it and it's stayed in a nice Jell-O-like state, you can actually, with permission from your parents, dissect it open. So just kind of cutting it open, the plastic, you can take a look at all the organelles again. And something I really like to do is use it as an opportunity to teach someone else. If you have younger siblings, this is a great way for you to become the teacher. So you can teach them what all the different beans represent, why there's spaghetti in there, why it has this feel to it. So really great additional activity that you can do. All right, excellent. So I'm going to leave this here, and then I'm going to bring you back over here. All right, so the last thing that we want to do is we want to finish up that chart. Again, that was on the website on the resources tab if you want to complete one as well. So let's just, I'm going to share again my screen, and we're going to just complete that last column of the table. Okay, all right, so let's think about the easy ones that I had mentioned right away. So the DNA, what was the DNA represented by in our model? String. And the string went inside the nucleus, and the nucleus was represented by the plastic ball. And just so it fits, I'm just going to put ball. All right. What about the cell membrane? What was represented by the cell membrane? The plastic bag. All right. Very outside of our cell. What did all of the organelles, what were they represented by? All the different beans and pasta. Don't forget that we had spaghetti in there as well, beans. And then specifically talking about one of those organelles, the mitochondria. Does anybody remember what specific bean I said I like to have represent the mitochondria? It's going to be the kidney beans. Kidney beans. Oh, a little bit outside our box, but that's okay. All right, and then we have our last two. So the cytoplasm, what gave that cell that feel that the cytoplasm would have? And that was the gelatin, right? And then I like to have the nutrients be represented by the water. Remember, we had to add the water in order for all of this to work. All right, excellent. Looks good to me. All right, so I'm going to stop sharing. All right, so thank you so much for joining us. I hope you've been enjoying joining us for DNALC Live. Don't forget that we do go live a few times a day, so check out our schedule at our website, dnalc.cshl.edu. What you can do is if you create one of these, feel free to share your pictures with us. We're on Instagram, Twitter, Facebook. So it'd be really cool to see what you come up with and what different materials you use in order to create your cell. All right, thank you so much for joining us. Feel free to subscribe, like, and have a great day. Thanks so much.